Hi, so in this video we'll be looking at the angiogenic switch. On the left hand side of the screen you can see the tumour cells, and on the right hand side of the screen you can see the nearby capillary to the tumour. However, the distance between them is not close enough for oxygen to diffuse across. This means that tumour cells at this stage of their development are in hypoxic conditions. Hypoxic cancer cells stabilize hypoxia-inducible factors, also known as HIF proteins. You can also see that separating the cancer cells from their capillary is the extracellular matrix. Hypoxia-inducible factors trigger the synthesis and release of pro-angiogenic factors such as vascular endothelial growth factor, or VEGF. However, as you can see here, VEGF is becoming sequestered by the extracellular matrix, meaning it cannot reach its target receptors on the endothelial cell membranes of the capillary. Therefore, cancer cells recruit macrophages and mast cells from the nearby stroma. These cells release matrix metalloproteins, or MMPs, and these proteins are proteases which have the ability to degrade the extracellular matrix. VEGF is now free to bind to its receptor on the endothelial cell membranes of the capillary. This has several effects. Firstly, it increases the permeability of the endothelium. This means that factors are more likely to be released from the capillaries. One example of this is the release of more matrix metalloproteins and urokinase plasminogen activators, or UPAs, which further degrade the extracellular matrix. This releases more VEGF, which can bind to its receptors on the endothelial cell membranes, and this creates a positive feedback effect. The breakdown of the extracellular matrix also releases other pro-angiogenic factors. For example, basic fibroblast growth factor, transforming growth factor beta 1, platelet-derived growth factor, angiogenin, and finally, angioprotein. Another role of VEGF is that it acts via the MAP kinase signal transduction pathway to cause endothelial cells to proliferate, and also the matrix metalloprotein-induced extracellular matrix breakdown activates integrins on these proliferating endothelial cells, allowing movement. Pro-angiogenic signals also recruit endothelial progenitor cells from the bone marrow, and these accelerate the process of vascularization, as you can see here. The tumour is now vascularized and has an adequate blood supply so it can continue to grow. I hope this video was helpful. Thank you.